Organelles excluded from the endomembrane system include Golgi bodies, endoplasmic reticulum, chloroplast, vacuoles. Endomembrane system consists of organelles like nuclear membrane, endoplasmic reticulum, Golgi bodies, vacuoles, and plasma membrane because they function in coordination. Here Golgi bodies, endoplasmic reticulum, and vacuoles are the part of endomembrane system. So chloroplast is excluded from the endomembrane system. The endoplasmic reticulum is composed of spherical vesicles, tubular and cisternal membranes, microfilaments, microtubules. Endoplasmic reticulum is composed of tubular and cisternal membrane. Which compartments touch endoplasmic reticulum cleared? Periplasmic and nuclear, luminal and extraluminal, cytosolic and nuclear, matrix and stroma. Endoplasmic reticulum cleared, luminal and extraluminal compartment. The space inside the membrane of endoplasmic reticulum is the luminal part, and outside the membrane is called extraluminal compartment. Rough endoplasmic reticulum is identified by the presence of lipids, ribosomes, DNA, chlorophyll. Rough endoplasmic reticulum consists of ribosomes in its membrane. Smooth endoplasmic reticulum lacks lipids, ribosomes, membrane, cisternae. Smooth endoplasmic reticulum lacks ribosomes. Due to absence of ribosomes, it appears smooth. Rough endoplasmic reticulum is abundant in cells that perform photosynthesis, secrete proteins, store starch, undergo meiosis. Rough endoplasmic reticulum is abundant in cells that secrete proteins because rough endoplasmic reticulum consists of ribosomes and ribosomes help in the synthesis of protein. Which is synthesized in smooth endoplasmic reticulum in animal cells. Enzymes, starch, steroidal hormones, DNA. Smooth endoplasmic reticulum in animal cells mainly synthesize lipids, and steroid hormones are lipids. Golgi apparatus was discovered by Robert Hooke, Camillo Golgi, Singer and Nicholson, Sladen. Golgi apparatus was discovered by Camillo Golgi. The cisternae of Golgi are typically tubular, flat disc-like, filamentous, spherical. These are the cisternae of Golgi, and these are flat, disc-like and parallel. This is the cis phase of Golgi apparatus, which is convex and is also called as forming phase, where it receives materials from endoplasmic reticulum. And this is the trans phase of Golgi apparatus. This exits material from Golgi body. The arrangement of cisternae in Golgi is random, parallel stacks, circular, helical. Cisternae in Golgi is parallel stacks. The cis phase of Golgi is described as concave, convex and forming phase, destructing phase, far from the nucleus. The cis phase of Golgi is described as convex and forming phase. It receives materials from endoplasmic reticulum. A cell specialized in secreting antibodies. Which organelle will be most abundant? Smooth endoplasmic reticulum, rough endoplasmic reticulum, lysosome, mitochondria. As we know, antibodies are protein and ribosome synthesis protein. So here, rough endoplasmic reticulum, which consists of ribosomes, will be most abundant. A toxin blocks lipid synthesis. Which structure is most affected? Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi cis phase, lysosome. As we know, smooth endoplasmic reticulum mainly synthesize lipid. So, smooth endoplasmic reticulum is most affected. If ribosomes detach from endoplasmic reticulum, it transforms into Golgi, vacuole, smooth endoplasmic reticulum, lysosome. It transforms into smooth endoplasmic reticulum. A mutation disrupts vesicles formation between endoplasmic reticulum and Golgi. Which process fails first? Protein secretion, glycolysis, ATP production, cell division. Vesicles help in transportation of protein from endoplasmic reticulum to Golgi. So here, protein secretions process fails first. Golgi cystine collapse and lose polarity, which process becomes defective. Protein sorting, ribosome binding, lipid oxidation, DNA replication. Golgi cystine helps in the process of protein modification, packaging and sorting. So here protein sorting become defective. Rough endoplasmic reticulum is involved in protein synthesis. This is correct. Ribosomes present in the rough endoplasmic reticulum help in protein synthesis. Rough endoplasmic reticulum has ribosomes attached to its surface. This is also correct. And due to the presence of ribosomes, it helps in protein synthesis. So here both assertion and reason true and reason explains assertion. Question number 18. Smooth endoplasmic reticulum appears smooth under the microscope. This is correct. Smooth endoplasmic reticulum lacks ribosomes. This is also correct. Due to the absence of ribosomes, it appears smooth. Here, both true and reason explains assertion. Golgi apparatus is also called as the forming phase. This is correct statement. The convex cis phase receives material from endoplasmic reticulum. This is also correct. That's why this is called as the forming phase. So here both assertion and reason true and reason explains assertion. The endomembrane system excludes mitochondria. This is correct. The endomembrane system consists of organelles like endoplasmic reticulum, nuclear membrane, Golgi bodies, vacuoles, plasma membrane because they function in coordination. Mitochondria contains their own DNA and ribosome. This is also correct, but this does not explain assertion. So here option B is correct. Golgi cisternae appear stacked. This is correct. They are disc-shaped membrane-bound sacs arranged parallelly. This is also correct. Due to their parallel arrangement, this appears stacked. So here option A, both assertion and reason true, and reason explains assertion. Match the endoplasmic reticulum type with its function. Rough endoplasmic reticulum is responsible for protein synthesis due to presence of ribosomes. Smooth endoplasmic reticulum synthesize lipid. Endoplasmic reticulum lumen is due to intracellular compartment formation. Match structure with discoverer or description. Golgi apparatus, Camillo-Golgi, rough endoplasmic reticulum, ribosome stud, smooth endoplasmic reticulum, smooth and lipid synthesis. Match organelle with its role. Cis phase of Golgi is receiving vesicles from endoplasmic reticulum. Trans phase of Golgi, sorting and packaging. Rough endoplasmic reticulum, synthesis of protein. The two phases of Golgi apparatus are identical and separate, different but interconnected, same in function, functionally inactive. They are different but interconnected. The principal function of Golgi apparatus is ATP synthesis, DNA replication, packaging materials, lipid breakdown. The principal function of Golgi apparatus is packaging materials. Vesicles from endoplasmic reticulum fuse with the transphase matrix cis phase nucleus. Vesicles from endoplasmic reticulum fuses with the cis phase of Golgi apparatus. After fusing with the cis phase, vesicles move to the chaperone proteins, trans or maturing phase, lysosomes, vacuoles. After fusing with the cis phase, vesicles moves towards the trans or maturing phase. The Golgi apparatus remains closely associated with mitochondria, smooth endoplasmic reticulum only, endoplasmic reticulum, ribosomes. Golgi apparatus remains closely associated with endoplasmic reticulum, both smooth and rough endoplasmic reticulum. Modification of endoplasmic reticulum synthesized proteins occurs in nucleus, Golgi cisternae, lysosomes, ribosomes. Modification of endoplasmic reticulum synthesized proteins occurs in Golgi cisternae. Golgi apparatus forms DNA and RNA, glycoprotein and glycolipids, lipase and protease, steroid hormones. Golgi apparatus form glycoproteins and glycolipids. The lipids and proteins synthesized from endoplasmic reticulum comes to Golgi apparatus, and here Golgi apparatus combines carbohydrates to protein and lipids to form glycoprotein and glycolipids. Lysosomes originate from mitochondria, chloroplast, Golgi apparatus, smooth endoplasmic reticulum. Lysosomes originate from Golgi apparatus. Lysosome contains photosynthetic pigments, hydrolytic enzymes, structural proteins, ribosomal RNA. Lysosomes, also called as suicidal bag, and it contains hydrolytic enzymes. It helps in breakdown of proteins, lipids, carbohydrate, nucleic acid, and waste material. Lysosomal enzymes functions best at neutral pH, acidic pH, basic pH, high temperature only. Lysosomal enzyme function best at acidic pH. Lysosomes digest all except proteins, nucleic acids, lipids, chlorophyll. Lysosome contains hydrolytic enzymes and it helps in digestion of proteins, nucleic acid and lipids, but not chlorophyll. Chlorophyll is the pigment present in plant cell. Vacuoles contain only water, shape and excretory products, ribosomes, DNA. Vacuoles contain cell shape, which is a solution of salt, sugar, and other solutes, and excretory products. The vacuolar membrane is called endomembrane, tonoplast, mesosome, cristae. The vacuolar membrane is called as tonoplast. Vacuoles in plant cells may occupy 5%, 20%, 50%, 90% of cell volume. Vacuole in plant cells may occupy 90% of the cell volume. Transport across the tonoplast is mostly passive, facilitated, active, osmotic. The transport across tonoplast is mostly active and it requires energy because the concentration inside the vacuole is more than the intracellular space. That's why the molecules moves from the low concentration to high concentration or against the concentration gradient, which requires energy. Contractile vacuole in amoeba help in photosynthesis, osmoregulation, respiration, cell division. Contractile vacuole in amoeba help in osmoregulation. Number of mitochondria in a cell is constant, is zero in all cells, varies among cells, always exactly 1. Number of mitochondria in a cell varies among cells. Mitochondria typically have diameter of 0.2 to 1.0 micrometer, 2 to 10 micrometer, 0.01 micrometer, 100 micrometer. Mitochondria typically have diameter of 0.2 to 1.0 micrometer. A cell has very high protein secretion, which organelle pairing would be most prominent. Smooth endoplasmic reticulum and mitochondria, rough endoplasmic reticulum and Golgi apparatus, lysosomes and plastids, vacuoles and smooth endoplasmic reticulum. A cell has very high protein secretions, which means it has more ribosomes. So here, rough endoplasmic reticulum and Golgi apparatus would be most prominent. Golgi apparatus help in protein sorting, packaging, and transport. A mutation reduces acidic pH inside vesicles, which structure loses function. Vacuoles, Golgi, lysosome, mitochondria. As we know, lysosomes functions well in acidic pH. So here lysosome loses function due to mutation. A unicellular organism living in fresh water would require which special vacuoles? Food vacuole, contractile vacuole, gas vacuole, storage vacuole. A unicellular organism living in fresh water would require contractile vacuole. Golgi cyst phase is damaged, which process is immediately affected? Protein secretion, steroid synthesis, ATP production, DNA replication. If Golgi cyst phase is damaged, it cannot receive protein. So, protein secretion process is immediately affected. A cell stores huge quantities of ions, whose structure helps maintain concentration. Nucleus, tonoplast, crista, chromoplast. Tonoplast, which is the covering of cell vacuole, this structure helps maintain concentration. Lysosomal enzymes act best at acidic pH. This is correct. They contain hydrolytic enzymes like lipases, proteases, carbohydrates. This is also correct. But this region does not explain assertion. So here both true and reason does not explain assertion. Vacuoles in plant cells can occupy up to 90% of cell volume. This is correct. Tonoplast regulates entry of ions and materials into the vacuoles. This is also correct. But this does not explain assertion. So here both true, but reason does not explain assertion.